guys, welcome back. This is the last of the cell biology unit. We're going to look at movement through a cell. So we talked a little bit about the plasma membrane, its functions. Let's talk about how things move through it. And there's a couple different ways that happens. So our main focus today is going to be looking at how things are able to move in and out of the cell. What are the processes of that happening? Really, really important for the cell to be able to do this efficiently. Otherwise, the cell's not going to be able to survive. So looking at this is very critical to the success of the cell. So we need to be able to explain why the process of diffusion, passive transport, and active transport are so important to the cells. Life cannot function unless things are able to move in and out of the cell, just like how we can't function if we can't take things in and out of our bodies. Additionally, you're going to look and be able to tell me a little bit about different types of solutions and what will happen to a cell if we put them into the cell. So should be very, very cool to be able to take a look at that and see kind of how those things work. All right? Any questions? Sounds good. Let's move on. So there are three different types of transport that we're going to be looking at. There's diffusion, there's facilitated diffusion, and there's also active transport. So we got quite a few different things that we're going to be looking at here. All three different types of transport are really important to the cell, so we're going to look at each one of them specifically. And the first one is diffusion, and diffusion is something that you should be very familiar with. Diffusion is what happens when you take a drop of food coloring and put it into some water. You watch it diffuse over time. We go from a high concentration to a low concentration. So just like when you put that droplet in there, we start off with a high concentration of the food coloring. But over time, because of the motion of the water, the movement of the water on the molecular level, the food coloring tends to spread out. It balances itself out. The same thing happens with different particles that occur both inside and outside of the cell membrane. You see in that first picture on the left, there's a high concentration of molecules on the left and very few on the right. But the other picture, it's all diffused out. So there's an even number of particles on both the left and the right side of the cell membrane. So diffusion is just movement of molecules from high to low concentration. Requires no energy, no extra energy, that is. And something to keep in mind is that it doesn't just stop. Movement of particles does not just stop because they're equal on both sides. This is what's called dynamic equilibrium. So even though on the right-hand side, there's no net movement, that is, that there's no more on each side, there's the same amount still going to be on each side, but particles are still moving all around. So I don't want you to think that just because it evens itself out, the movement stops. That's not the case. It's just that the movement from one side to the other is equal to each other. That's where we get the term dynamic equilibrium from. So diffusion, high to low, no extra energy, and pretty simple, pretty straightforward. All right? Next one is facilitated diffusion. And again, we're moving from a high concentration to a low concentration, but this requires a protein channel. All right, so these are molecules that can't directly diffuse across the cell membrane. So these may be things like ions. It's the same thing. It's from a high to low concentration, doesn't require any energy, and it just simply requires a protein for them to be able to transport through. So sometimes these protein channels are open, sometimes they're closed. It just depends upon what's needed inside the cell, what's needed to go inside and what's needed to go out. So definitely some really, really important things going on here with regular diffusion and facilitated diffusion. Again, the main difference is that facilitated diffusion requires a protein channel. Both are from high to low concentration. All right. The big difference here, we have what's called active transport. When you think of the term active, you should be thinking things like energy. You know, get really excited. Active requires energy. So this transfers from low concentration to high concentration. It goes against the concentration gradient. So that would be like that drop of food coloring in that water going from this low concentration where it's all mixed within the water all the way back and taking it out and forming that droplet that you started with again. So it's very, very difficult. It requires a lot of energy for this to take place. All right? Again, it goes against the concentration gradient and requires energy. It requires ATP for this to happen. One example of this is the sodium-potassium pump. Very important in a lot of life processes, including things like muscle contraction and nerve firing. So keep in mind Active transport is the one that's really different because it requires energy and it goes from a low concentration to a high concentration. It goes against the concentration gradient. It goes against what would naturally happen. All right? So it's just something to think about as we go through this. Exocytosis and endocytosis. Some things are just too big to be able to even go through protein channels. And that's where exocytosis and endocytosis come into play. Exocytosis is reserved for large molecules that are simply just leaving the cell. Exo to like exit. So large molecules leaving the cell, and endocytosis is when we take large substances into the cell. And if you notice how this works is that the membrane tends to pinch off a vesicle. So as you see, as a material either goes into the cell or goes out of the cell, it tends to pinch inward. All right, so if the molecule's going into the cell, it's going to pinch inward and kind of trap that molecule inside what's called a vesicle. All right, it's made of the phospholipid bilayer of the cell membrane, so they can fuse with the cell membrane and be a part of it as well. When things are performing exocytosis, the vesicle just fuses back with the cell membrane and releases whatever it wants to the outside. So this is for larger molecules that can't quite make it through the protein channels. All right, so just some things to think about. Endocytosis and exocytosis are for large substances only. One thing that's also really important to understand is the movement of water in a cell. And the movement of water in the cell is called osmosis. Osmosis is just simply the movement of water to equal out solute concentration. So if we look at this picture here, we have a selectively permeable membrane, just like the cell membrane, but there's a higher amount of the purple dots on the right side than on the left side. So the concentration is higher on the right than on the left. Well, what happens is that since we can't move the purple dots to the left side to even out the concentration, we have to move the water to the right side. And what that does is that evens out the concentration, the number of purple particles per amount of water. Yes, there are still more purple particles on the right side, but there's more water to lower out the concentration. All right, so this movement of water is called osmosis. You've probably heard the term osmosis before, which is used to filter out chemicals and things like that in water systems. So when we look at osmosis, you want to keep in mind that water is always going to move towards the higher concentration of solute, the higher salt concentration, to equal it out. So the way I always teach this, or the way I always think of a good way to remember this, is salt sucks. Salt sucks the water out, so whichever has the higher salt concentration, that's the direction the water is going to go. And we have three terms to describe the solution that a cell is being placed in. We have what's called a hypertonic solution. Remember, all these refer to the solution, not the cell. So that's a really good way to help you remember that. Hypertonic, meaning we have a high solute concentration on the outside. So if we have a higher concentration on the outside than the inside, water will move outside the cell, right? Water is going to move outside the cell. If we have more salt on the inside than the outside, we have a hypotonic solution. More salt on the inside than the outside means that water moves into the cell. And the same salt on the inside and the outside is isotonic. So that doesn't mean that the water doesn't move, but it means that it moves at the same rate. All right, and that's what provides a normal, healthy cell. In a hypertonic solution, the cell tends to shrivel because water leaves it. In hypotonic, the cell seems to get too large because water wants to move into it. So we can see that hypertonic, hypotonic, and isotonic are all referring to the solution in which the cell's been placed. And as long as you remember salt sucks, it should really help guide you towards figuring all of this out. All right, so hopefully you understand diffusion, passive transport, and active transport, and understand that diffusion and passive transport, or facilitated diffusion, are don't require energy and go from high concentration to low. Active transport is low to high, requires energy, and goes against the concentration gradient. And then also predict the effects of a hypotonic, hypertonic, and isotonic solutions on a cell. So we got a lot we covered today. We'll look at some more of these a little bit later on, and then we got one more unit to go before the end of the first semester. All right, guys, have a good day.